Why is hemoglobin electrophoresis the main test for sickle cell? Imagine trying to find a tiny needle in a haystack. Hemoglobin electrophoresis acts like a special magnet that helps doctors spot that needle, which in this case is the abnormal hemoglobin causing sickle cell disease. This test is the main way to diagnose sickle cell because it can accurately tell the difference between normal and abnormal hemoglobin types. Hemoglobin is the protein in red blood cells that carries oxygen, and everyone has a mix of different types. In sickle cell disease, the body produces hemoglobin S, which causes red blood cells to become sickle-shaped, leading to blockages and other health problems. Hemoglobin electrophoresis works by applying an electric current to a blood sample. As the current flows, the different hemoglobin molecules move through a gel or capillary system. Because each type of hemoglobin has a unique charge and structure, they migrate at different speeds. This separation creates distinct bands or peaks, which can be measured and visualized. The pattern shows how much normal hemoglobin, called hemoglobin A, and abnormal hemoglobin, like hemoglobin S, are present. This makes it possible to see if someone has sickle cell disease, sickle cell trait, or other hemoglobin disorders. One reason hemoglobin electrophoresis is preferred is its high accuracy. It can clearly distinguish between normal hemoglobin and variants, such as hemoglobin C or other abnormal types. This specificity helps avoid misdiagnosis. It also provides a percentage of each hemoglobin type, which is important because sickle cell disease usually shows a high level of hemoglobin S and little or no hemoglobin A. In contrast, sickle cell trait shows a mix of both. This quantitative detail helps doctors understand the severity and nature of the condition. Another key point is that hemoglobin electrophoresis is used in newborn screening programs around the world. Detecting sickle cell early allows for prompt treatment and management, which can prevent serious complications later in life. While initial screening tests like the sickling test or solubility tests can suggest the presence of sickle hemoglobin, electrophoresis confirms the diagnosis with certainty. Modern techniques like capillary electrophoresis and high-performance liquid chromatography have made the test even more precise and capable of identifying other blood disorders as well. In summary, hemoglobin electrophoresis is the main test for sickle cell because it provides a clear, specific, and detailed picture of the types of hemoglobin in the blood. This helps doctors diagnose the disease accurately, determine if someone is a carrier, and plan appropriate treatment. It is supported by guidelines from health authorities and remains a cornerstone in the diagnosis of sickle cell disease. If you or someone you know is being tested, always consult healthcare professionals for personalized advice and interpretation of results.